Excited for today's speaker, and we're glad that you're all joining us for this event. Today's speaker is Dr. Dom Esposito from Frederick National Laboratory for Cancer Research. Dr. Esposito is the director of the Protein Expression Laboratory and the project lead for RAS reagents. Dr. Esposito's group is responsible for generation of DNA, cell line, protein reagents in support of the NCI RAS initiative, a targeted drug discovery program focused on identification of therapeutics against RAS-driven cancers. In addition, the Protein Expression Laboratory produces proteins in support of other National Institutes of Health projects, including COVID-19 and other pandemic preparedness research. The laboratory also focuses heavily on invention and development of novel technologies for improving protein expression and production. Dr. Esposito received his BA in chemistry at La Salle University in Philadelphia and his PhD in biochemistry and biophysics at the Johns Hopkins University Bloomberg School, where he studied bacteriophage site-specific recombination. He then worked as a postdoctoral fellow at NIDDK, where he studied protein-DNA interactions involved in HIV viral integration reaction. And prior to joining the Frederick National Lab for Cancer Research in 2001, he worked for 3 years as a staff scientist in the protein engineering group at Life Technologies, now part of Thermo Fisher, where he helped to develop the gateway recombinational cloning system. For the past 6 years, he has also been the project lead for NF1-related research within the NCI RAS initiative. In this role, he has led the Frederick National Lab for Cancer Research team in a collaboration with researchers in the lab of Dr. Frank McCormick at UCSF to investigate the biochemistry and structural biology of neurofibromin, the goal of better understanding the role of the protein structure and function in NF1 disease. In addition to identifying the high-affinity obligate dimeric nature of neurofibromin, the Frederick team solved a cryo-EM structure of the neurofibromin dimer, which aided in identification of the likely biochemical mechanism of severe NF1 disease point mutations. NF1. So last year, the group has continued to explore details of the structural flexibility of neurofibromin and has begun to investigate potential therapeutic approaches to target neurofibromin directly. Today, Dr. Esposito will talk about biochemical and structural analysis of neurofibromin to elucidate mechanisms of NF1 disease and potential therapeutic options. I will now stop sharing my screen and will hand over the floor to Dr. Esposito.
All right. Thanks, Kalyan. Just want to thank the Gilbert Family Foundation for giving me the opportunity to present our research today. So I'm going to start with a little background to sort of let everyone know a little more about the Frederick National Laboratory for Cancer Research, which I imagine some people probably haven't heard of before. So FNLCR is the only one of the U.S. national laboratory system that's dedicated exclusively to biomedical research. So it's funded out of the Department of Health and Human Services by the National Cancer Institute and is operated by a contractor known as Leidos Biomedical Research. And the mission of FNL is really to provide a unique national resource for the rapid development of technologies to address urgent biomedical problems for the U.S. And that includes, obviously, cancer, HIV/AIDS, and now emerging infectious diseases. And so we're located in Frederick, Maryland, which is in the foothills of the Blue Ridge Mountains, where this cow is located, sort of equidistant from Baltimore and Washington, D.C., for those not familiar with the area. In 2013, when Harold Varmus took over as NCI director, he was asking questions about, well, how do we use this national laboratory resource, which we've had here for a while, to do something important for cancer research? And the idea that he came up with was applying FNL resources to what at the time was considered a drug discovery program on the undruggable oncogene RAS. And so the first FNL national mission, as it was called, was developed, and this is the NCI's RAS initiative. And I'll talk a little more about the goals of this in a couple slides, but really it's a three-pronged approach to understand RAS biology, develop therapeutics specifically to target mutant RAS proteins, and to produce and distribute reagents to the external community that works on RAS research to sort of enable the extramural community as well. So we started in October 2013. One of the first things that we did was to hire Frank McCormick from UCSF as our scientific advisor to the program to help sort of develop the ideas behind the programs that we were going to work on. We expanded pretty quickly over the next few years, hiring a lot of staff. We're right now about 75 FTEs in this program for the most part. And along with hiring really good personnel, we've acquired a lot of new technology over these years to advance our abilities to do screening, structural biology, optical imaging, and mass spectrometry. And I'll talk a little bit about some of the impacts of these later. The program was originally renewed in 2018 for 5 years, and we're in the process now of going through a re-renewal process to continue working on some of these projects. So I probably don't need to go into a lot of detail with this audience about the RAS family of small GTPases, but just to highlight that, the target of this program is are the three main RAS isoforms that play a role in human cancers. Those would be KRAS, NRAS, and HRAS. And really, our program is very heavily focused on KRAS because of its role as a main driver for a lot of really bad cancers. And all of these proteins, of course, have very similar G domains, the actual GTPase portion of the protein, and differ mainly by these hypervariable tails, which are post-translationally modified and are involved in interactions with the membrane. And each of these different isoforms has a slightly different membrane association and probably a slightly different location on the membrane. But as I said, the main goal for us is really KRAS, which is the driver for almost all pancreas cancer, about half of colorectal cancers, and about a third of lung cancers. That represents, in data that's now probably a few years old, it's probably more than this now, about 138,000 cases per year in the US, new cases, and over 1 million new cases annually across the world. And some of these are obviously some of the worst cancers in terms of survival rates. I know these numbers are a little bit better now than they were 10 years ago when the slide was made, but these numbers are still probably in the single digits for pancreas cancer and still fairly poor for the rest of these. Now, I've struck through this line that was on this slide originally because when the RAS program started, and even when we were renewed in 2018, there were no currently approved therapeutics for treatment of RAS-driven cancers. That has changed over the last few years with the approval of 2 therapeutics, one from Amgen and one from Mirati, to treat specifically G12C driven RAS cancers. So that's primarily lung cancer. But I would say those are relatively new. And the initial data on those, while they have some efficacy, it doesn't look like at this point that either one of those is likely to be a long-term major benefit to patients. There's a lot of issues around resistance to those drugs, and there's a lot of development still going on. The good news is that there are probably 20 or so drugs that are currently in the clinic or going into the clinic. So I think there's a lot of opportunity in the next few years for targeted RAS therapies. So again, just briefly to go over this for anyone who's not as familiar with the full RAS pathway, the basic understanding of how the RAS activation occurs in normal cells is that stimulation by growth factors of receptor tyrosine kinases in the membrane lead to the activation of these proteins known as GTPase exchange factors. So SOS is probably the canonical one. The role of the SOS protein is basically to remove a nucleotide from the RAS protein. In this case, it's the inactive RAS GDP bound form. And by removing that GDP, it allows RAS to rebind GTP, which is found at much higher concentrations in the cell than GDP. So releasing the GDP and allowing GTP binding turns RAS into its active form bound to GTP, which allows it to interact with downstream effector proteins such as RAF kinase, and that leads to the signaling that you want for normal pathway activation with growth factors. So proliferation, growth, differentiation, progression through the cell cycle. Under normal circumstances, when those growth stimulation signals are stopped, this nucleotide exchange process is stopped, and we allow RAS to hydrolyze GTP, which is what it does as a GTPase, and that reverts RAS back to its inactive GDP-bound form. So RAS itself is a really poor GTPase. The GTPase activity is really low. And so in order for this to be efficient, this process is stimulated in cells by another family of proteins called GAPs, or GTPase activating proteins. And of course, this is where neurofibromin is the main player in shutting off the RAS GTP state and returning it to the GDP-bound form to sort of complete the cycle. In oncogenesis with RAS, what we see are a variety of mutations in the RAS protein, which do one of two things. Either they completely eliminate the ability of RAS's natural intrinsic GTPase to work. And so RAS is stuck in the GTP-bound form because it cannot hydrolyze GTP. Or there are mutations which abrogate the ability of GAP to bind to RAS and mediate the stimulation of its GTPase activity. Either one of those have the same effect of basically locking RAS into the GTP-bound form, leading to uncontrolled MAP kinase signaling through RAF. And of course, you get the same exact effects that you get normally. It's just these are now uncontrolled and continuous proliferation and differentiation, and those lead to oncogenesis. So either of those two mechanisms can really be at play here. And this is kind of the scheme that we went into the RAS initiative thinking about in terms of how do we target specifically these oncogenic mutants of RAS, which are no longer able to hydrolyze GTP or are no longer able to interact with GAP proteins. So to do this, we built a number of capabilities into the RAS Initiative program, sort of modeling after the kinds of things you see in biotech or pharma companies that do drug discovery. We have a really strong basis in protein science driven by biochemistry and biophysics, hardcore structural biology using all of the modern methods from X-ray crystallography to NMR to cryo-EM, and then groups that develop biochemical and cell-based assays to allow us to do screening for compounds. Underpinning all of that is a RAS reagents group that generates all of the materials that are used by these different internal groups, and also can supply these to external laboratories, either collaborators or just other extramural investigators who are working in RAS. And then finally, we developed a sort of full-fledged drug discovery program, including medicinal chemistry, computational chemistry, and a variety of different means to develop our own RAS inhibitors using a number of different processes I'll discuss in a couple of minutes. So the other area that was really important in the development of this program, and was one of the visions that Harold had when he set this up, was that the RAS initiative would not be just a uniquely NIH-based process. It would be what he called the hub and spoke model. So the RAS initiative would be the hub centered at the Frederick National Lab, but we would form these spokes out to various collaborators out in the community. And these would be a combination of academic collaborations, strategic collaborations with other government laboratories, and through a specific mechanism called a contractor collaborative research and development agreement, even could extend this to interactions with for-profit companies. And so over the first 10 years of the program, we've developed a large number of collaborations with the various entities listed on this slide, a number of academic institutions around the world, a number of different government laboratories, including other national laboratories, and then again, these C-Creator relationships we have with companies. And these can range from interactions where we provide assays that we've developed that can be used to screen libraries, which these companies have, or they can be sort of full-fledged collaborations where companies support a number of FTEs at the Frederick National Lab, and we work on these projects in collaboration, sharing the intellectual property, and leveraging the ability of these companies to get drugs into the clinic. So we have a variety of different levels of collaboration with the different companies. So overall, just briefly, the goals of the program, really the first one was a better basic understanding of KRAS biology and biochemistry, and how it interacts with its various effectors at the membrane of the cell. So I think the key point here is that KRAS only activates RAF kinase when it's at the membrane. It doesn't do this when it's just floating around in solution. And understanding the molecular mechanism of that RAF activation by KRAS was one of the initial targets of our program. And the key to that is that a lot of the published work in this area and even the drug discovery on KRAS was always done with soluble KRAS that didn't have that membrane-associated tail. So we were really interested early on in leveraging our technologies to make these really complicated proteins to generate sort of real KRAS that could bind to lipids and bind to membranes and explore the potential differences between lipid-bound KRAS and sort of soluble KRAS. And as part of that, we've developed a collaboration with Lawrence Livermore National Lab in order to use their high-performance computing systems to help us predict some of the interactions of RAS with the membrane and RAS with downstream partner proteins, and using their computer modeling to sort of highlight the kinds of experiments we could do and then iterate back and forth to try and understand these mechanisms better. And that's something that we are still working on currently. So the second goal, I think, which was one that was the initial real focus of this program, was to actually develop assays and identify compounds that we could use to inhibit oncogenic KRAS. So remember, when this program started, there were a few companies that were working on RAS drugs, but it was a pretty thin area because people really didn't think this was a very druggable target. Fortunately, I think other companies have worked on this a lot and have shown that, in fact, RAS is definitely a druggable target, and that gives us a lot of opportunity now to look at different mechanisms for targeting. So in addition to some of those partnerships I talked about, which really involve a lot of high throughput screening, which are done by companies who have large libraries, or fragment-based screening, we also have our own in-house drug discovery efforts that are focused heavily on covalent tethering approaches. So these are approaches in which molecules are targeted to cysteine residues on the proteins, and we've generated our own proprietary cysteine tethering library at FNL that has more than 2,000 compounds, and we've used that to attempt to identify novel pockets on the surface of KRAS by mutating the entire surface of the protein to add cysteine residues and identify where some of these compounds might target. And so that's a fairly major effort for our internal drug discovery process. And then just to highlight that we actually we do have 2 likely drug candidates that were developed from the RAS initiative in partnership with BridgeBio, which we hope will enter the clinic sometime later this year. And these are fairly novel targets. So one of them is a G12C inhibitor that hits the GTP bound state, unlike the GDP bound state targeting molecules that are out in the clinic already. And a second one, which is a compound which is capable of disrupting the RAS protein kinase PI3K alpha interaction. So the final area that we're really interested in, of course, is the generation of reagents to enable the larger community of RAS researchers. And so this is a big focus of the program as well. We've distributed a large number of plasmids and cell lines from the FNL and through repositories like AdGene pretty much across the world. And we've generated a number of materials that we license out as well to companies to enable their research. And as part of this, we like to leverage the technologies we've developed as part of this program to really help elucidate the role of other players in the RAS pathway. And this is really what started our work in the NF1 arena as an area that we thought we could apply some biochemical, biophysical, and structural studies on using the technologies that we developed, and maybe come up with ideas for other drug discovery targets within the larger RAS pathway. So that's basically the background of what the RAS initiative is focused on. I want to turn now to discuss in a little more detail our research in neurofibromin. And this really is the example of the it takes a village approach, because in addition to work done by our team at FNL and the RAS initiative, we have a great collaboration with the McCormick Laboratory at UCSF and a number of other partners who've played major roles in different parts of this process. And so there are a large number of people involved in this research. So I definitely don't have to, I think, discuss with this group the background of what neurofibromin is and why it's relevant, except to remind everyone that this is a very large protein. It's a 320 kilodalton gap, which is much, much larger than most of the other gaps that are out there. Its role is really that we know of is to stimulate the low intrinsic GTPase activity of RAS, and defects in this gene obviously lead to NF1 disease with a variety of different phenotypes. And I think the thing that was most interesting to me starting out in this area was that there are a wide variety of genetic alterations that lead to NF1 disease. So unlike RAS, where basically you have a very small set of oncogenic mutations that lead to RAS-driven cancers, there are literally mutations all throughout the NF1 gene, point mutations, truncations, frame shifts, loss of allele issues. So there's a lot of complexity for a protein that really only has a very tiny part of it that we know has actual activity in the context of the RAS pathway. And so as a biochemist, that to me was a really interesting thing to look more into detail about. The initial work in the RAS initiative focusing on neurofibromin really focused specifically on the interaction of RAS and GAP, the GAP domain of NF1. And so our structural biology group, led by Dhirendra Sumanshu, was the first to determine the binary structure of KRAS and NF1 gap in the ground state configuration, as shown in this data from this 2019 paper. We were really interested in understanding a little more about how some of these oncogenic mutants of KRAS are able to abrogate the binding of NF1. And so some of the structural data was, I think, particularly informative. Sumanshu's team went on to develop a more interesting structure, which is the ternary structure of the KRAS G domain bound to the GAP domain of neurofibromin and the EVH1 domain of the adaptor protein SPRED1. So I think the most interesting take home from this was, if you look at the model here, the binding of KRAS to the GAP domain looks pretty much identical here as it did in this binary structure here. So KRAS GAP interaction is pretty much the same. What's interesting is that the EVH1 domain of spread, unlike some original arguments which had suggested that it might bind to KRAS directly or to the GAP domain, it turns out that the EVH1 domain binds exclusively to this sort of dark green domain here, which is what we call the GAP-EX domain. And if you look at the linear structure of neurofibromin, that GAP-EX domain exists in two parts, one at the N terminus and one at the C terminus of the GAP domain. So although these two are separated by several hundred amino acids in primary sequence, the folding of this portion of neurofibromin actually leads this GAP-EX domain to form a single structural domain, which is responsible for binding to SPRED1's EVH domain. And the really nice thing about this structural data and the analysis that Sumanshu's team did on mutants is that we can explain a lot of the results from various disease mutations in SPRED1. In particular, the impact of phosphorylation of the S105 in EGFR mutants, which eliminates spread NF1 binding, and as well as a number of Legius syndrome mutations in SPRED1, which are very nicely explained by their presence on this border between the binding of the GAP-EX domain and the EVH1 domain. So this was part of this research involved us trying to get a handle on the activity of the full-length neurofibromin protein. And so we set about to try and generate some of that protein, thinking this would be a pretty challenging task for a protein this large. As it turns out, we were really fortuitous that in our insect cell-based expression system, full-length neurofibromin is actually relatively easy to produce at fairly high level and is readily purified to homogeneity at yields that are really quite good for a protein this size. And so that got us thinking we might have some of the techniques and technologies to answer the question, what exactly does the rest of this protein do? Because we know it has this GRD domain that we have structure for that has GAP activity, and we know it has a SECPH domain, which presumably is involved in binding to the membrane. But beyond those two regions, there's really not a lot of clarity as to what the rest of the protein does. There were domains sort of listed in the literature as defined by interacting proteins, but a lot of those don't really have good biochemical conformation, and it's not entirely clear what the role of those specific interactions are. So the majority of this protein, we just don't really understand what it does and why mutations in those regions might have an impact. And so that's one of the areas that we decided we would focus on. So initially, the investigation of full-length recombinant neurofibromin led us to do some biochemical and biophysical analysis. And we were surprised to find that, for the most part, this protein is what we call an obligate dimer. So it exists only in dimeric form at pretty much any concentration that we can measure, and certainly any concentration that we see of this protein in cells. And we can see this by a number of fairly simple biochemical techniques like size exclusion chromatography, where it runs at a size appropriate for a dimer, but also a lot of advanced techniques like small angle X-ray scattering and mass photometry, which are generally much better at measuring low concentration molecular weights. And the gold standard here really is analytical ultracentrifugation. This technique really gives us an accurate size of the protein down to very low concentrations. And in this case, predicts that the dimerization affinity of neurofibromin monomers is somewhere less than 1 nanomolar. So suggesting that really at all concentrations that we can test, and likely all concentrations in the cell, this protein is an obligate dimer and doesn't exist as a monomer. And we were able to confirm these results in cell-based assays, thanks to work done by our colleagues in the McCormick lab, in which they took differentially tagged NF1, one with a V5 tag and one with a flag tag, and expressed those in HEC293 cells that lack endogenous NF1. And when you co-express these two proteins and pull them out with an anti-flag antibody, what you see in the third lane here is that in addition to the flag-tagged NF1 protein, you also pull out the V5-tagged protein. So again, clearly showing that these two proteins are functioning as dimers in the cell. So using this information, we decided to go on and try to get some structural data. So we generated a number of negative stain EM images. We had about 42,000 particles, which gave us this sort of peanut-shaped molecule here that looked like it was very dimeric when we just sort of eyeballed these. And you can see some nice side views here as well that give you an idea of the shape. And using this data, we were able to construct a 20 angstrom negative stain model of the protein. And this is data that we published in our late 2019 paper in JBC, shown here. So the take home from this paper, which has a bunch of other studies of various domains of neurofibromin, was that likely the dimerization was represented by both intermolecular and intramolecular interactions. It was a very complex folding process. And we thought maybe that had something to do with how you can get point mutants all over this gene to contribute to NF1 disease. So the one mistake we made in this paper was in looking at these images, we sort of assumed that the two halves of this protein were split along a symmetry axis here in the middle, and we colored these monomers, the green lobe at the top and the blue at the bottom, just like the two lobes of a peanut. As it turns out, as you'll see in a minute, that's actually not the structure of this. It's much more complicated than even we expected from here. And the way that we were able to determine that is to move this from these early negative stain EM studies into more advanced cryo-EM studies in collaboration with the team led by Jana Anjenovic at the FNL National Cryo-EM program. And Jana was critical in developing the structural data that we generated. I'm going to show you some of that data here. Just to highlight what I'm sure most people here know, 2022 was a good year for the production of NF1 cryo-EM structures. There are now a total of 4 of them from 4 different groups, and I'll highlight a little bit in a minute some of the similarities and differences, but I'm going to show you mostly the data that we generated from the FNL structure here. So Jana's group was able to take the protein that we provided. They did some extra cleanup to get it to work a little better in cryo-EM. And ultimately, they were able to take about 250,000 particles and develop a 3D cryo-EM structure. The initial structure, although highly resolved at the central region, you can see had sort of these red images on the end here show slightly lower resolution. And so they were able to use a number of computational techniques to refine this structure to the final structure that we have here shown as just 1/2 of this particular molecule, where we now have resolution down as low as 3.3 angstroms, and overall the local resolution is really strong. So it gives us a pretty good image of what's going on. So The overall structure of neurofibromin is what's termed a lemniscate homodimer. So it's basically a figure 8 shaped protein. And I'm going to just highlight here the regions that we sort of know about in terms of structural data from crystallography. So in purple here, the GRD, the gap-related domain, and in pink, the sec-pH domain. So those are sort of the known parts of the protein before these structures are available. The rest of this was pretty much unknown territory. So if we drop down to a more atomic resolution here where we can see the actual alpha helices, what you'll see is that the body of the protein is made up pretty much entirely of alpha helices. So this was somewhat predicted by previous notions that these were heat-like repeats. These domains are actually not really canonical heat-like repeats, but they are large helical bundles, basically, that make up the entire middle of the protein. And you can see the dimeric structure here. One monomer is shown in gold and starts right about here, runs through this portion of the protein down to here, and then the second molecule in silver runs here. So there's sort of a side-by-side dimer. The other thing that's really obvious from looking at the structure is that the reason for the obligate dimerization of this protein, it has an extremely massive dimer interface in the center of the protein, burying a huge amount of hydrophobic surface area in the center here, which probably leads to the majority of the dimerization power. But in addition to that, it actually has strong interactions between the N-terminus of one molecule and the C-terminus of another. And of course, that happens at both ends. So that's sort of an extra layer of hydrophobic glue that holds this protein tightly together. And so this is a very, very large surface area for a dimer, and other proteins that share similar amounts of surface area, such as mTOR, also have very high dimerization affinities. So one really interesting thing that came out of our work, as well as the work from Andrew Ellisden's lab in Australia that had the first published structure, was that this configuration of neurofibromin that we see in our structure is what we call a closed state or an autoinhibited state. And the reason for that is because if you overlay this cryo-EM structure with the ternary structure that I showed you earlier from Samanchu's group, if you map the GRD on top of our structure here, then the spread EVH1 domain is sticking out here into solution, and that's fine. That's perfectly reasonable. But the RAS protein from that structure is completely buried inside of the rest of neurofibromin. So our cryo-EM structure is incompatible with RAS binding to the GRD and suggests that we have a structure here which is occluded from RAS binding and is likely to be non-active in terms of activation of RAS. So the two other structure papers that were published last year, the first one from Caroni, which was published in Nature, and the second from Tim Mayer's group and Klaus Schefsek, which was published in Molecular Cell, both have structures which show a second form of neurofibromin. In the case of Caroni's paper, it is one in which one side of the protein seems to have a closed configuration similar to what we see, and a second one in which there's been a rotation that has exposed the gap domain in a way which would make it competent for RAS binding. In the case of the Schefsek and Mayer structure, they actually see two open domains where both GRDs are exposed, one here and one over here. And again, both of those could potentially bind RAS. These two papers differ in their argument for what leads to the activation of the protein. The Caroni paper suggests that it has to do with stabilization of the closed state by a zinc ion in the structure, whereas the Schefsek and Meyer paper suggests that it's a nucleotide binding that stabilizes it. We don't see either of these in our cryo-EM structure, although we have enough density in those regions to in theory, have seen both of those. So this is something we're still investigating to try and understand what the differences are between these two models, which actually are not compatible with each other because the residues that are involved in binding zinc are actually the same ones that are involved in binding nucleotide. So this is an open area of investigation I'll touch on a little bit later. But just to summarize, again, we have 4 structures. Resolutions are very similar amongst them. Some of them are better than others in certain places. There's very different activation mechanisms suggested. And all of these papers have some really nice supporting data that help to define the roles of different parts of this protein. Unfortunately, for the field in general, all of these structures suffer from the exact same problem, and that is they are missing significant regions of density. And those regions include some really vital loops in multiple different domains of the protein. And so I will mention in a few minutes why this is a big deal and what we think we need to do to get around this problem. Because although we have this nice structural data, it still hasn't told us a whole lot about the activation of the protein and the role of some of that structure in the gap activity. So what can we do with the structure that we have? What can we learn from this? And I think one of the key things we're really interested in is the potential use of this structure to explain genotype-phenotype relationships in NF1 disease. And this is work that was published just a couple of weeks ago in this PNAS paper, is collaboration between my team at FNL, Frank's team at UCSF, and Jana's team at the National Cryo-EM program. And so I'm just going to highlight the ways that we think we can use the structural data to inform genotype-phenotype. This came out of work that was published in 2018 in the American Journal of Human Genetics, which identified a series of point mutations in NF1 in this domain that we call the B domain between 844 and 848, which led to severe NF1 disease. So we decided to take a look at some of these mutations using our recombinant protein system to see if we could make these proteins and learn something about what was wrong with them. And we were really surprised to find that it was extremely difficult to make any of these proteins in our insect cell system. So in normal insect cell expression of wild-type neurofibromin, we produce a lot of protein, as shown here by this lane in the band labeled T, which is total protein. And all of that protein is completely soluble. So when we lyse the cells and spin out all the debris, all of the neurofibromin is in the supernatant, in the wild-type protein. In contrast with these point mutants in these 844 to 848 regions, we see good expression of the protein, but we see absolutely no soluble protein. So all of this protein is insoluble in the cells. And for those of us who work in insect cell protein expression, that's a real sign that there is a defect in the folding of these proteins, which leads to targeting for those proteins for degradation. The insect cell process is viral and lytic. And so the first thing the virus does when we are infecting these insect cells to make protein is shut off the proteasome. So any proteins that are targeted for proteasomal degradation and polyubiquitinated basically form these giant aggregates in the cell and they just precipitate out. So this is a real canonical sign that there is a serious protein folding defect in these cells with these mutants. And we see this when we express even smaller bits of the protein. This is just the left half of neurofibromin, which again, we can express beautifully in normal cells, but these mutants produce almost no soluble protein. So the team from Frank McCormick's lab, led by Lucy Young, did work in cells to sort of compare the data that we had in recombinant protein. So here, these are proteins that are expressed in HEK293 cells that were knocked out of wild-type NF1. And when Lucy expresses wild-type flag-tagged neurofibromin in these cells, it looks great. However, all of these mutants basically produce very low levels of protein. And when they're analyzed for ubiquitination, you can see that all of them are heavily ubiquitinated. Another sign of some significant defect in protein folding is the upregulation of HSP70 seen in all of these cells as well. So again, these are suggestions that there is a problem, a defect in the folding pathway. So if we look into the structure a little bit more of this 844 to 848 region, you'll see that it sits in this yellow helix here, which is part of a 4-helix bundle, which are colored red, yellow, blue, and green here. And that bundle represents a tightly packed series of helices, which sits underneath the gap domain, which is shown here in purple. If you sort of look in the plane of the screen up at the end of this, you'll see in this picture in the bottom right here that the center of this helix is packed with hydrophobic residues, and that forms the core of this hydrophobic bundle. Another way to look at this is with a hydrophobicity plot shown up here, where the more hydrophobic residues are colored in red. You can see the center of these helical bundles is pretty much all red residues. So not only do these residues from 844 to 848 fall into this region, But we see a number of residues on these other three helices, which are also hydrophobic and in the core of the structure. And if we look at the phylogenetic relationship between human, mouse, rat, and Drosophila neurofibromin, we see that all of these residues inside this helical bundle, which are shown in the yellow highlights here, are very well conserved over all of these proteins. And the ones which are shown in red stars here are actually ones which are known to cause NF1 disease. So there clearly seems to be a link between disruption of these helical stacks here and an actual phenotype in the disease. So to really nail this down, we went and looked at some of these other patient mutations in these regions, in these other helices other than the yellow one. And in addition to trying to express the proteins that have the actual disease mutations, we decided to make additional mutations, which we considered mild mutations. So as opposed to changing a hydrophobic to a charged residue, which most of these mutations over here are, or a helix breaking residue like proline, we decided to substitute them with other hydrophobic residues to, in theory, create a mild change which should not disrupt the helical bundle if we're right about the argument. And in fact, what we see with recombinant protein is indeed in these cases where there's these red disease mutations, we again see a complete loss of soluble protein when we make these proteins in insect cells. Whereas in the case of the mild mutants, we generally rescue the ability of those proteins to be made. And the in vivo data shows very similar results, where again, these patient mutants give either very low levels of neurofibromin or no detectable amounts, and we can rescue a lot of that with these mutations. So the model that we think best explains what's going on here is the concept of what's called subunit poisoning. So the idea being that there are normally two alleles of neurofibromin, and so there are four possible types of dimers you can make: homodimers of allele 1, homodimers of allele 2, and then heterodimers of the mixed alleles. And if we have one variant, one allele, which is producing a destabilizing variant, which is polyubiquitinated and targeted for degradation, that's going to impact three of those four possible dimers. And only a wild-type dimer would actually give you functional neurofibromin. So whereas you might think that a mutation of one allele would lead to a protein level of 50%, this would argue that it is more likely we're going to only see somewhere in the 25% level of functional protein. And this may represent a major mechanism of protein insufficiency with point mutants that don't necessarily affect gap activity, but do disrupt this very complex helical bundle structure of neurofibromin. And I think it's really nice because this is very consistent with data published a number of years ago by Corinna Anastasaki and David Gutman's lab, which showed that there were a number of mutations and truncations of neurofibromin, which led to a significantly reduced amount of protein expression, a lot more than you would think if you just lost an allele. So for instance, this NF1-9, which is a premature stop nonsense mutation at residue 2207, leads to a protein level down around 15%, which would be very hard to explain if this protein was just effectively non-functional. So instead, I would argue that these proteins and many of these truncations are competent to interact with wild-type monomers and are then being targeted for proteasomal degradation, poisoning the wild-type protein that's available. So I think this really holds up well and is very consistent with the data that we've obtained. So where do we go from here with regard to biochemical and structure function studies of neurofibromin? So we have these cryo-EM structures, but like with a lot of structural biology, they really open up more questions than they necessarily have answered. So we still don't have a clear idea of what's activating this protein from that closed state to the open state. Is it metal? Is it nucleotide? Is it something else like phosphorylation? In order to look at this, we're developing a number of different biochemical and cell-based assays that we hope can be used to monitor binding of neurofibromin to other proteins as well as gap activity. And then we're in the process of generating site-directed mutants to test a number of these activation hypotheses and explore what some of the roles of these parts of the protein might be. We're also very interested in additional genotype-phenotype studies based on both the work I just described on these helical bundles, but also in trying to identify some disease mutants which maybe don't affect degradation of the protein and don't affect gap activity, to sort of argue maybe what some of the other parts of these proteins might be impacting. And of course, we are continuing our structural biology work, now expanding it into the structure of signaling complexes. So neurofibromin bound to RAS, bound to spread, possibly KIT and other partner proteins. To assist in this, we've developed some specific affinity reagents, both monoclonals and Fabs, against some of the regions of the protein that are flexible in the hopes of stabilizing those and learning more about those regions. And then I just wanted to highlight in a couple slides, I think one of the more interesting take-homes of this, which is these very large unstructured loops in what we call the F domain. So this is the C-terminal part of neurofibromin that extends from somewhere around 2300 to the end of the protein at 2818, depending on which isoform you're using. This region of the protein has a large number of predicted phosphorylation sites. So this is from phosphosite, and you can see that all of these balls are references that show phosphorylation at particular residues. And you'll see that there's a cluster of them here and another cluster at the end. And these red bars at the bottom are not just to differentiate these clustered phosphosite mutations. These are actually regions of the structure where we have no structural data from any of the 4 cryo-EM structures. So likely these are large unstructured loops that are involved in interactions with other proteins. And I think it's particularly interesting to note that this long bar here, which covers about 140 amino acids, if you look at the endpoints of this at 2450 and 2615, they are represented in our structure by these red balls. So this is the end of our structure, and then it picks up over here. Those are spatially separated in the cryo-EM structure by about 7 angstroms. And yet this is over 160 amino acids of space. So this loop could literally be extending anywhere in this structure. It could be extending out to this side of the protein. It could be easily extending over here to the gap domain, or it could be binding other proteins that are interacting with neurofibromin. We really have no idea because we can't see any of that structure at all. So one way that we hope to explore this a little bit more is using a cross-linking based approach. So this is cross-linking mass spectrometry, in which we take a cross-linking reagent like the one shown here. This happens to be BS3. This reagent will bind to and covalently cross-link lysine residues, and it contains a spacer arm that sets a very specific length to how far that cross-link can occur. So the spacer is about 7.7 angstroms, which means the lysine to lysine distance is probably around 13 angstroms. So if we have 2 lysines in the protein that are 13 angstroms apart, we can cross-link those, and using mass spectrometry, we can identify where those cross-links are. And this map here of the full-length neurofibromin protein, each of these purple lines is a cross-link that we detect with mass spectrometry on our recombinant full-length neurofibromin. And so you can see this region over here on the right is where that loop is that I just described. And we see interactions of that loop with a large number of different parts of this protein. So there are interactions with the far end terminus. There's interactions with parts of the 400 to 600 region, as well as interactions with parts of the gap domain, suggesting that this region is flexible and has the ability to interact with a number of different portions of the protein. And one way to really sort of visualize this and where we hope to go in the future with this is to map onto the structure. So here you're looking at one monomer of neurofibromin in orange and the other one in blue, and we're sort of looking at a side view. Each of these green balls represents the position of a lysine residue, which we see an observed crosslink at. And they're connected by these silver tubes, which represent the crosslink. And so you can see some of them, like this one down here at the bottom, there's 12.2 angstroms between them. That's pretty reasonable for these cross-linking reagents. That's likely a very real cross-link between 2 lysines in this region. But you can see up here that we have lysines, which we have placed, again, on this missing loop, which we've just modeled into the structure randomly here because we have no idea where it is. Those lysines in that loop region are interacting with a variety of different parts of the protein. So one over here, some down here, some into the middle of the protein here. And you can see that these lengths are completely unrealistic. So we cannot measure a crosslink of 129 angstroms, which means simply that these lysines are not really in this location. They have to be located in other regions where they're able to be close enough to these lysines here to crosslink. So we hope that we can computationally map the options for where this loop may be. And we can engineer additional lysines into the loop in different locations to try and map out the overall structure and really understand where this loop is residing and what it might be interacting with. And that's going to be the target of some of our structural work going forward. So finally, on the last slide, I just want to mention that we're really interested in taking some of these ideas that we've learned from the structure and considering potential therapeutic approaches that we might use. So one of them would be the idea that maybe we could stabilize these mutant neurofibromin proteins that are being degraded. This could be something as obvious as proteasomal inhibition, which I know people are already working on, but also the concept of maybe developing chaperone proteins that might be able to stabilize these unfolded helical bundles and prevent those proteins from being degraded. And the other area that I think we're interested in within the RAS initiative that relates to neurofibromin is the idea of generating stabilizers of NF1 and RAS. And the idea here is that we know from structural data that RAS interacts with the NF1 gap domain, as shown here in the green and cyan structures. And that binding to RAS occurs at the exact same place that RAS binds to other effector proteins. So SOS1 in the gold and RAF in pink. And so the idea is that if we could lock KRAS into an interaction with the NF1 gap domain, then we might prohibit its ability to signal downstream to RAF or to SOS. And the idea here is that there is a pocket in this interaction, which was identified computationally by our computational team based on the structural data that I showed earlier. And the hope is that we could design some kind of a glue molecule that might be able to bind these two together and lock RAS into this unproductive interaction with NF1 and then abrogate downstream signaling. So these are all areas that we're considering for the next sort of iteration of our drug discovery programs. So again, this work is done by a very, very large number of people across multiple sites. All of the cryo-EM structural work was led by Jana Janović's team at the National Cryo-EM program. They've been really helpful in generating this data and helping us understand what's going on. There's a large team at UCSF that is run by Lucy Young and Frank McCormick's lab, who generated all of the cell-based data that I showed you. And then a number of RAS initiative teams, both on the protein side in my group, as well as the collaborations with the biochemistry and biophysics group in Andy Stevens' lab and our great structural biology team led by Dhirendra Sumanchu. And so all of those people were really vital in developing the work that I showed you. So I am more than happy to stop here and to take any questions. Stop sharing here. All right.
Dom, that was a terrific talk. I have a question that's probably a little naive, but it seems that a lot of the mutations prevent the stabilization of the dimer. But what if, we had been discussing earlier, whether a mini gene version of NF1 could have the function without all the extra stuff on it. And so the question is whether a kind of a truncated version of NF1 that doesn't concern itself with kind of stabilizing this very large molecule, but just sort of has the business end on it, would rescue the GTPase activity?
Yeah, that's a good question. I mean, I think certainly under some circumstances that's true. I mean, we know that the gap domain alone can in some cases sort of rescue the activity. I think the bigger question is whether there's a regulatory role that neurofibromin is playing in addition to this that is not just the gap activity. And I mean, obviously that has to be the case. You're not going to make a protein this big and a gene this big if you don't. But yeah, I mean, one of the things we're really interested in exploring right now is trying to understand this activation mechanism. And is the control of neurofibromin just to regulate on and off in different parts of the cell cycle or in different locations or different cell types, in which case maybe the gap domain alone or some smaller piece is enough to fix a lot of these issues. So yeah, I think that's a good point.
Okay, thank you.
Maybe I can go. I'm not sure if anybody else has had their hand raised. My name is David Solomon. I'm a cancer geneticist and brain tumor researcher at UCSF in San Francisco. Thanks for the very interesting talk, looking at the kind of neurofibromin protein function and domains. One of the kind of particular questions that I have is focused kind of on the genetics, and why do you speculate that basically all pancreatic cancers have KRAS mutations, and lots of colon cancers and other things have RAS mutations, but basically virtually never have NF1 mutations? And then when we look at gliomas in the brain, they essentially never have RAS mutations and are, very frequently have NF1 gene inactivation. And why do you think there's selection pressure in gliomas and nerve sheath tumors for NF1 inactivation versus acquiring RAS mutations in these epithelial type cancers?
Yeah, I mean, I would love for somebody to answer that question, because actually, as a completely non-geneticist, non-clinician, I've always wondered the exact same thing. It is remarkable, in fact, that we don't really see, NF1 mutations in cancer. They're generally not at all like that. But I do not have any answer for that question at all. It's a great question. There's clearly some regulatory role that's different. But yeah, no, sorry, that is. I would love to know that answer. Yeah.
Yeah. Do you think it suggests that there are kind of other than RAS functions of NF1 inactivation that gliomas are trying to, and gliomas and nerve sheath tumors are trying to select for, versus these epithelial cancers that acquire RAS mutations just want RAS as opposed to the kind of more pleiotropic effects of NF1 inactivation? Or
that could be. I mean, certainly with the RAS mutants that we see in cancer, I mean, most of them are gap independent. So at that point, it really doesn't matter. I guess they're never a second hit on NF1 wouldn't matter, right? Because they don't even work with gaps. But the reverse question is a good one. Why in the gliomas do you not get the RAS mutations having the same impact? So I'm sure there are other functions of this protein. I think that's going to be the most interesting questions to ask, because what else is it doing? It's got to be doing other things than this.
I have a quick question. Hey, thanks for the great work and the great talk. And my question is with regards to open and closed state of NF1. And I'm not sure if this has been published, so I figure if I haven't read it, but what's the percentage of NF1 open to closed state? Or is there a range? Can it, like, for instance, can it be tied? Can we increase the amount of open in the wild?
Yeah, so those are great questions. That's exactly what we're interested in finding out. So the problem here is that this definition of open and closed state is done by cryo-EM. So you're picking particles, and so it's actually unclear in some of these papers just how many of the particles they had in the open state versus the closed state. For instance, we do think we see some open state in our data, but not enough for the folks in the cryo-EM group to be able to build a model of it. So it's not entirely clear. There's no biochemical data right now that says anything about how much is in one state versus the other. We don't even really have very good assays to measure this. So that is definitely one of the goals, is in a normal prep of this protein, how much of it's active, how much of it's not, and can we force it one way or the other? But again, I think the biggest issue right now is trying to nail down what the activation mechanism is, because both of these papers have a really good set of data that suggests that they know what the activation mechanism is, but they are completely incompatible with each other. So we're trying to sort of assess what the activation process is, because if you could do that, I agree, locking it into one state would be that would be a great argument to try.
Dom, great presentation, great work. You've really done a tremendous amount to elucidate, the structure of the neurofibromin protein and really help us all generate even better ideas. So one of the things, as you know, we still struggle with assays for measuring functional neurofibromin. I was just wondering, I mean, you share that that your group are in developing new assays. Is there anything you can elaborate on that we could potentially adopt across multiple labs if you're close to validating something that would help us?
Yeah, so we're, right now we've been focused really on biochemical assays for our recombinant protein work. so we have a biophysics group that does a lot of assay development, and they're working on a number of different binding assays to look at interactions of neurofibromin with other proteins, but also higher throughput gap assays that we can use with recombinant protein. In cell-based assays, that's definitely something we're going to look at. We have started working on some Brett assays, but those again are more about interactions with other proteins rather than measuring the gap activity in cells. So I don't have a good answer, and I think I'm in the same boat. I think we'd like to think about better ways we can do cellular assays. It's probably worth having a larger discussion about that sometime, because I do think we need some ideas for that.
Great. Thank you.
Hi, Dom. This is
Hey.
from MD Anderson. This is a great talk. Thank you. I realize the dimerization data is very interesting and explains why like David's group is seeing 25% protein left in some of the mutants. One other thing in that data set is that there are some mutants that result in 75%, like around 3/4% functional protein. Like, I'm wondering if you have explanation for that one.
No, I'm not sure. That data is really nice from a sort of bigger qualitative perspective. It's not been clear to me whether you can really accurately tell the difference between 50 and 75% in some of that data. So I've always kind of hesitated to take home that 75 was a solid number. We haven't looked at those proteins that had the higher levels. We have actually, in collaboration with Karina, looked at a lot of those other mutations in that paper and been able to confirm that the ones that have low levels of protein, actually we can mimic that with the recombinant protein. So it's actually a lot more data than I showed, but we haven't looked at those ones that were higher. So that would probably be a good idea. I think we could probably get more quantitative data with recombinant protein and see if we can understand that. Because I can't really hand wave a good argument for why you'd see above 50% in those cases. But yeah.
Thank you.
Yes, we are at 3:04 p.m. now. If there are no other questions, I would like to announce, first I would like to thank Dr. Esposito for this wonderful seminar presentation. And I would like to announce the next one, which will be given by Dr. David Solomon, who is in our audience today on the 10th of May, same time at 2 o'clock in the afternoon, Eastern Standard Time.
Thanks. Looking forward to.
Yep. Thanks everybody for coming.
Bye everyone. Great talk. Thank you.
Thank you.